Hi.
Hello. Can you hear me well?
Yes. Just yes, I could hear you.
Okay, just let me hit the last things. Okay,
I can't hear you now. No, I can't hear you. Are you able to hear me? Okay, no, I can't hear. I was able to hear you, but now the voice is gone.
what about now?
Yes, now better.
Okay,
Yeah, now
let
I
me
can.
just, okay. Okay, just a second. I will close the door because I'm in a library. Okay, should be fine, right?
Yes, yes.
Okay, so I will share the screen. Do you see this?
Yes.
Okay, so this part is actually the introduction and this outline. Okay,
Yeah.
so I focus more on the strategies of unravel the true effect of VUSs. Okay, so and I will
Yes.
give just a heads up on giving examples about how to go on Nomad, how to check everything, how to go for population database, giving some examples. uh After that, frequent literature mining and oming search, explaining what is oming, how to go for it, like expression databases, how to look into those prediction tools, giving some example in like from individual prediction to comprehensive prediction, the use of AI, and then strategies like functional assays. And in the functional assays, I will give a brief description of what are the most common on the most common functional assays, describing the role of splicing, why we are interested in that, how to apply for therapeutics, giving again some example, focus on splice AI. And then if we have time after this one, that will be the part of the high throughput splicing assay.
Okay. So, because you mentioned that you wanted us to collaborate with the others with their VUS. So if you could just say that, we are working on it together, like you're helping me analyze my variants, and maybe in the near future, like I can pool all the VUSs, something like that.
Of course. Can you still hear me well?
Yes. Yes.
Okay.
So we could at the end just briefly discuss about that so that we can just put it across to the group that what we are doing and how we can increase the number of USs from all of them for analysis. So
Yeah.
in the initial introduction also I can just mention that probably. So you and we were together in the Netherlands for your PhD, is it?
Yes.
Oh.
So I was like, like I was in Italy for university, and then I went to Radboud University Medical Center for PhD, 5 years, and then after graduation, I moved to Marseille.
Okay.
And in between, there was like this experience in Tel Aviv, in which there's another center for human genetics, but this one was more for inherited hearing loss.
Okay.
But since Usher 2A is a gene that is involved in deaf blindness, I was working in helping them with Usher 2A related things.
Okay.
It was just, it was a small fellowship that I got it, and it was in between the two work experiences. Yeah.
Or no? Okay, I'm just going to be back in 5 minutes.
Sure. Thank you.
Hi, Ria. So Dr. Riccardo has logged in, and we just checked his audio and video, so it seems to work.
Okay, perfect.
Hi, Ria.
Hi, good evening, Dr. Riccardo. Looking forward to your talk and nice to meet your acquaintance as well.
Thank you. Please call me Rick. I'm not like a doctor, just like I'm a scientist, like
Yes.
it's fine. Like here, if you say doctors, they think you're a medical doctor, so which I'm not.
No, on the contrary, this particular group that we have has a mix of everybody. So it has ophthalmologists and it has scientific doctors and even genetic
Okay.
counselors like myself. So it's basically everyone getting to try to understand genetics a little bit better.
Of course. No, it's I'm very grateful to Damaji for the opportunity.
And if I may ask, how did you and Dr. Tanmay meet each other? How do you know each other?
So
Yes.
I had this gene CFAP, right? CFAP as a VUS.
Mhm.
So I was just asking, we have a common friend who, when I was doing my fellowship in Belgium, she was there. She's from Greece, Dr. Evangelia. So I just asked her about my query if she knows anything, and she told me that he is her colleague and friend, and he's working with that gene, so to consult him. So that's why we just got in touch.
That's great.
Yeah.
I think we'll have a lot of Q&A. I have a feeling we'll have a lot of Q&A in this session.
Yeah, I'm sure we'll be having. I think it'll extend.
Mm.
I mean, I tried to keep the presentation quite broad and easy to digest, but there are so many different contents. It's just I'm happy to, like if there are Q&As that I won't be able to respond on everything, I'm very happy to try to find another occasion and probably to focus on specific topics, in the next useful occasion.
Yes, most definitely. We can, I mean, given how much time you can spare for us, we can turn it into a series of 2 or 3, how much ever like we seem to have the requirement for. Yeah. Mhm. So it's 8:30. We'll just wait for 2 more minutes for more people to log in and then we can start.
Yeah. Okay.
आ गया याद।
Ria, should we start? Because otherwise it will extend too much.
Okay, so shall I share the screen and the presentation?
Yeah, just a minute. Let Ria just join back. Ria, are you there? Should we start? Hi, Shelja.
Hi, Tanmay. Hello, Ricardo.
Hi,
Thank
hi,
you so
Dr.
much
Shasha.
today. Looking forward to your talk.
Thank you.
I think let's begin because
Yeah, Tanmay, yeah, begin.
So hello and welcome to the 13th session of our ocular genetics interest group today. We have a accomplished PhD geneticist, Dr. Riccardo Sangermano from Boston. A brief introduction. He has completed his MSc in medical biotechnology from the Federico II University in Naples, Italy. Here he had joined the group of Dr. Nicola Brunetti Pieri at the Telethon Institute of Genetics and Medicine, where he worked on congenital syndromes and inborn errors of metabolism, in particularly lysosomal storage disorders and MPS type 1. In 2013, he was awarded a Marie Curie Early Stage Research Fellowship as a part of the ITN Research Training Network. And he joined the Radboud University Medical Center in the Netherlands for his PhD. During those years, he contributed to the identification of novel genetic variants of IRDs, with special interest in ABCA4, in the unraveling the missing heritability. I think he's going to speak about it today. After obtaining his PhD summa cum laude in the medical science from Radboud University, he joined the Ocular Genomics Institute of Massachusetts Eye and Ear Infirmary at Boston. As a postdoctoral research fellow, his current research focuses on investigating and functionally characterizing photoreceptor essential genes and genetic modifiers of disease severity in IRD patients. So thank you, Riccardo, for agreeing to take this session for us, and over to you.
Thanks. Can everyone see the screen?
Yes, it's visible.
Okay. So, hi everyone, and I'm very glad to be here. Thanks, Tanmajid, for inviting me. Today, as I promised, I will just give a brief introduction of giving some examples and some introduction of what I'm currently working on, with a special focus on variant interpretation in genetic eye disorders. As Tamerji already mentioned, I come from Italy. I worked there till the first years after my university. Then I had to move to the Netherlands, and I'm now based at Massachusetts Eye and Ear in Boston. And it was very interesting to see that during this timeline that starts from 2006 till 2025, now we have genetics. The world of genetics has accomplished so many things, and I was very lucky to live and train in a period in which so many exciting things happen in terms of population genomics, databases, and expression databases, and also the first gene therapy for retinal disease, but also like new prediction tools that finally assess splicing effects for genetic variants. So I felt like I need to train during this period. So as you may probably know, the inherited retinal degenerations in IRDs in short are genotypically and phenotypically heterogeneous. You can see here from this Venn diagram in which each circle represents a species, and these are the genes that are responsible when mutated of each specific disorders. And as you can see in these diagrams, some of the genes are at the intersection of multiple diseases, and one among all is ABCA4, which is the frequently affected gene in IRDs and is responsible for a different type of macular degeneration like Stargardt disease, so cone dystrophy, cone rod dystrophy, and also retinitis pigmentosa. All these are different clinical phenotypes of different severity. So one important thing to before starting with the presentation is that especially for recessive disease and recessive IRDs, for what concerns my experience, the effect of each variant needs to be studied in details, because each variant and the effect of each variant contributes to the clinical phenotype. And ABCA4 is one of my main these. So it's like I just want to give an example about the ABCA4 genotypes. So of course you have normal phenotype in absence of mutation, but also in presence of any type of variant, even severe, because it's like a recessive disorders. But then depending if you have combination of severe and mild, or severe, or two moderates, or severe and moderates, you might experience different degrees of severity, ranging from late onset Stargardt with onset around the 40s, the 50s, and on to the called now intermediate Stargardt, like a sort of standard textbook clinical phenotype. And then it's very rare, we're talking about 15 to 5% cases that biallelic combinations of ABCA4 severe variants or severe plus moderate variants can give rise to more complex phenotypes. So it's important to know now that with the advancement of the sequencing technologies, we have so many variants that are unclassified, and overall, like, going under the umbrella term of a variant of uncertain significance, or VUS, and this is due to the tremendous advancement in sequence opportunities. Nevertheless, we really need to classify those variants. There's a tremendous urge to classify those variants, either out as benign or likely benign, or as in this scheme, to move to the classification of pathogenic and likely pathogenic, and this for several reasons. The two most important of which are, of course, we want to give a full molecular diagnosis to our patients in our clinics, but also most patients, if their genotype won't be fully clarified, they won't be eligible for any therapies, and any gene therapy or any other, they will not participate in any clinical trial. So it's very important to classify the VUS and move them to pathogenic or likely pathogenic if possible. And another thing is that also most of these VUSs are non-coding variants, meaning variants either present between exons, so in the intronic region, or present in the regulatory parts of the gene at 5' promoter or 3' untranslated region, so the 5' or 3' UTR. And some other variants are even intergenic. These are the most complicated ones, and I won't be able to disclose in this presentation, but it has been shown that there are variants that are apparently not related to engine, because they are in genomic segments which are in between two genes in the, what in the region that before was called as junk DNA, and yet those variants are able to somewhat interfere with the expression of nearby genes. So today, this is like a brief outline of what is going to be my presentation about. So I will discuss all the main strategies to unravel the true effect of the VUS, starting with the consultation of either general population databases, so databases with just unaffected individuals, but also patient allele frequency databases, such as Clinicians. Also, I will discuss that it's very important to recurrently perform like literature training and OMIM search. I will give some example for that as well. I will discussing about how extra insights and information could be achieved by consulting gene expression databases. And finally, in the second part, I will talk about prediction tools, recent AI tools, and also functional assays, which are not available for every variant. I want to just start mentioning that, and I will explain you why. So starting from and please interrupt me for any question. I'm able to take your question either during the presentation or at the end. So talking about the population databases, it's very important to, when you have a VUS in your patient, it's very important to understand what is the situation of the presence of that variant in the general population. And to do that, the standard tool that, let's say, the gold standard tool that we are using these days is NOMAD. NOMAD stands for so it's a gene aggregation databases that, as you see, is available either for genomic assemble HG38, but if you have data that are in HG19, so it's like a GRC H37, is also available. And at the moment, like the most updated version of NOMAD is the NOMAD version 4. And this NOMAD version 4 is tremendously helpful because it contains genomic data from either genome sequencing or exome sequencing of almost, let's say, approximately 800,000 individuals. And as you know, most of the sequencing projects are mainly performed on European or European descended patients, but the important thing of one of the most important features of this latest version is that you now have finally a 30% of individuals that belong to other ethnicities, including African, East Asian, Middle Eastern, South Asian, and so on. So here I want to give an example. By the way, for each web page that I will be explaining today, you will always see the link here, and I'm able to provide this link after further questions. So let's pretend that I will be interested in looking in this gene, TMEM216. So now this is a gene that is associated with a syndromic form of retinal degeneration. Two forms in particular. One is called Joubert syndrome, and the other one is Meckel syndrome. And these two syndromes, they do have retinal phenotypes together with other clinical signs, including for Joubert syndrome, including like cerebellar dysfunction. And say that I want to type in before this gene, so to get more insight about what's the status in the general population. So this is actually the screen. It will appear this screen in which you have like all the information about the transcript isoforming. It will give you a small table in which will tell you the state of the art of how many synonymous means and loss of function are present and have been detected in the general population for each gene of interest, and also give you these important metrics, which is the difference between how many variants are present, for instance, how many loss of function stop variants are present compared to, so observed in the general population, compared to the number of variants that are expected. And this expected is a sort of extrapolation of the frequency of the mutation rate and the length of each gene. So sometimes it already, by looking at this number, if I have like a situation in which you have way less missense variants than expected, already it's an indication that that gene might be a dominant gene, or a gene that is like responsible for clinically phenotype when mutated. This is a coverage plot. It tells you that TMEM has like 5 exons and how they have been sequenced. So, like you can already have a high confidence that all the variants that have been present in the database are actually true variants, detected in patient, and a high read depth. And the most important thing that I want to discuss with you today is actually what happens if we observe like a specific metrics. For instance, I want to give the example of these two non-coding variants in TMEM. So these two non-coding variants are at the same position, minus 266. So we are talking about prime UTR, what is called promoter region. And as you see, they have been observed either 87 or 407 times in the in the general population, so the unaffected population. And yet you would expect that, especially for the second variant that is present 407 times, you shouldn't expect more than one homozygote, while instead you only see one. So already by looking at spotting those metrics with an indication for something that doesn't really follow the normal, the expected genetic frequencies of the variant. So if you use this database a little bit more extensively, and then you hit on the variant ID, you will go to another page in which you will see this variant present 407 times in what population has been found. And you will be surprised that majority of the variants are present in an African or African American population and completely excluded, not present, not found in a European population. So this already indicates that this variant, yes, is highly present in the not affected population, but it's actually present in a subset of the population, which is actually only the African and African American ancestry. So these are also admixed Americans, Americans that have at least one parent or descent from African America. And, some remaining are people with clear ancestry. But the fact that it's present so highly in African Americans and not in any European, given that the European are almost, they have over 1 million alleles in this database, it should be, it's for us like it trigger our attention. And at the same time, the other variant, this is the GT variant, but the G2A variant instead is prevalent in the South Asian population. As you see, there are 79 allele found in South Asia. And so we thought that this would be something interesting and worth like investigate. And in fact, once we performed like a functional assay, in this case, a promoter luciferase assay to study the function of the promoter, we observed that, okay, so first of all, I want to just give more explanation about what you figure A. So in figure A, you see the genomic region, and this minus 69 is just the same minus 266. It's just that this in a different transcript. Okay, so pardon me, anytime you will read the minus 69, I just mean the minus 266 position. So this is the position, and this is like the gene, okay, the TMEM 261. So by looking at the promoter, the predicted promoters, there are two putative promoters, one which is very close to the gene itself, and another one which is upstream. But we notice also that there are, if you use like other databases, there are predicted region of transcript regulation. For instance, here, which are ataxic region, and also these are traces that come from like conformational study using human retina, in which we know that this region here is predicted to bind the transcription factor CRX and the transcription factor OTX2. And we notice that this minus 69 or minus 216 was actually falling in the region of transcript regulation. So this was a nice thing to observe. And once we performed the actual functional test, we saw that for the wild type construct, in which there was the normal, the reference variant, you would have an expression level that's like the highest. And while you have the G2A or the G2T VUS, you will have this expression level that is reduced and is almost reduced, similar to a situation in which you do not have the promoter sequence P1. While instead, if you keep this P1 in the construct and you remove the P2, so the other promoter here, the putative promoter, expression stays the same. So in conclusion, we were able to, by just observing the frequencies of non-coding VUSs that were by the promoter and comparing to the distribution of unaffected patients in the general population, we're able to actually validate two promoter non-coding regulatory variants that they result in a lower expression of the TMEM And we got into those variants because we had a cohort of African American retinitis pigmentosa patients. They were all homozygote for this G2A change, while our collaborators, they had a cohort of Pakistani retinitis pigmentosa patients homozygote for the G2A. It was very interesting that although these two populations are apparently unrelated, like Pakistani Bangladeshi versus like Africans and African Americans, still the same position, just two different changes, either G2T or G2A, was able to decrease massively the expression of this gene and give rise to retinitis pigmentosa. And this was just, so the study is published now, and this is just the example of how you could interpret a VUS based on comparison between your patient cohort and the information provided by unaffected population database. So if we instead we want to move to having insights just by looking at sequencing data from patients, at the moment we are using two main databases. One is ClinVar, probably you're all familiar with, and the second one is LOVD. LOVD stands for Leiden Open Variation Databases. So they are quite overlapping, but I would say that I have quite a positive experience with LOVD, just because LOVD is actively curated and you find many more references and many more genotypes. So it's also relatively user-friendly. Once you open the LOVD, you just need to add the gene that you are interested in and click, and then will bring you directly to the main page. And today I will give you an example about ABCA4. So ABCA4, I mentioned before, is a gene associated with non-syndromic IRD, and in particular with Stargardt disease. So if you scroll, this is just adapted table from the LOVD data because the LOVD data is much more broad, and I just want to focus on a small subset of data. But if you look at the top four recurrent variant in ABCA4 in patients, you will see that the first three are missense variant, while the fourth one is a non-coding, but a position -10. So it's just -10 base pairs before the exon. And in this case, we are talking about exon 39 of ABCA4. So here you see the reported, the instances in which each variant was found in patients, okay? So it was very striking to observe that this non-coding variant was present in more than 1,000 patients, okay? But while the first three missense were classified as pathogenic, either mild or moderate severe or hypomorphic, this one was initially classified as benign. So we thought it was a bit odd, given the presence in patients, like the very recurrent presence in patients, but also because if you see NOMAD V4, again, like our population database, you see that this -10 theta C is actually present 800 times, okay, approximately 803 times. So it's very interesting to see that in over 1 million alleles, you will have in the general population only 863 instances, while in a other subset, smaller, that is only by ABCA4 patient, you will have a presence of over 1,000 instances. So that was for me trigger to study this variant functionally. And the way I did that was a combination of studying directly patient material, like patient-derived cells, and performing RCR on their transcript, If you do that, you will see that the variant is not benign, but actually causes like an exon skipping, in particular exon skipping of exon 39, but also exon skipping of exon 39 and 40. And this because the two exons are close together, like pretty close by. And so it's like by affecting the let's say the non-coding position and -10 of the exon 39, you're basically skipping exon 39 and 40. While instead, as you see in the regular control and in the retinal human retinal sample, you do not observe those skippings. And this is just the electropherograph shows the exon 38 to 40 boundary and exon 38 to 41 boundary. So first skipping event and second skipping event. So again, this was the example of you could reclassify a VUS, and in this case, actually reclassifying from a status of benign variant to the status of pathogenic. We thought that initially the variant was considered benign just because there were not enough studies about splicing. And usually people consider splicing variant the variants that are +1, +2. So the first two upstream and downstream each exon. While this was an example in which even positions are not canonical, so there are not +1 and 2 and -1 and 2, could still result in a severe phenotype. So now I will, after discussing the database, how to look into that and not find specific information just by comparing frequencies, ethnicities, and so on, I will move to another point, which is why we need to constantly check into literature, but also into specific database. And I will be introducing OMIM database. So the OMIM database is a database of human genes and genetic disorders. And I really recommend you to dig into this database because it's super useful. It's plenty of references and it gives a lot of information about the gene you are interested to. And again, I will give you an example. So if we type into our search line the search menu TM216, which I said before is actually associated with syndromic IRD, you will see that like after our work, the database has already changed and has included retinitis pigmentosa. Okay, something that was not present like less than 6 months ago. And it's nice that it's so frequently updated that if you click and you scroll all the information once you go on the phenotype, it already explains that in 71 patients from African and South Asian ancestry, they found, with non-syndromic retinitis pigmentosa and blah, blah, blah. So even though you have checked this gene once on these databases, I really recommend to do frequent searches. All the state of art of the literature is constantly updated, and you will always find new genotype associations. And while instead the second database that I would like to mention here is the database of gene expressions. And this is very important because if you have a VUS, maybe a VUS in a gene previously not related with retinal degeneration, one thing you would like to check is whether this gene, how expressed this gene is in the retina or in other tissues in case you have a syndromic disease. So at the moment, the state-of-the-art database for that is the GTEx. And the GTEx is also very user-friendly. It's a genotype tissue expression, GTEx. And here you just ask you to either insert the specific SNP ID if you have like a specific polymorphism, or just the gene. I usually insert the gene name. And again, back to the example of TMEM 216, you will see that it gives you the expression level in each human. And if you go to the database today, you will not find the retina data yet, the expression in the retina yet, but those data are still, so these data do exist and are part of, let's say, the group that submitted so one of this part of this GTEx consortium that provided this data as a subset of data that are only relative to the photoreceptor expressions. And I know that this data will at certain point included and integrated in the main plot, but at the moment they are not. But yet this is like a very useful tool for all genetic disorders, because you have like a mix of either like heart, liver, kidney organs, brain subsections, like parts of the brain. And I would say probably is actually almost complete except for the eye field. But I just want you to know that this data exists. And in fact, here I want to show you one figure, part of the same manuscript, in which we compare the expression of TMEM in peripheral retina in 411 samples versus the same expression in all tissues that you see here. And it was very interesting to see that we observed an increased presence and expression of the TMEM 216 in the peripheral retina compared to the sum of tissues. And I think this is an extra evidence of the relevance of TMEM 216 in the so not anymore a systemic syndromic disorder, but also non-systemic eye like retinal degeneration phenotype, like retinitis pigmentosa. So now, after database and predominantly just the information that they were already the parts of consortium, I will focus on prediction tools. So this is a sprint of one of our cases found solved for a biallelic combination of CFAP 4 and 10 variants. In particular, this subject this subject had a non-syndromic retinitis pigmentosa. He carried like a missense, cysteine 25 arginine, and a plus 91 non-coding variant. So I just want to highlight here that there are multiple prediction programs, and I would say that most of the programs are focused on missense variants, okay? So some of them I would say that are my favorite, like those are mostly used even in literature, are CUD and Revel. I invite you to look into the specific databases for those. The CAD score is significant above 20, and it, let's say, the higher the score, the higher is the predicted the seriousness of that specific change within evolution. And Revel is a similar thing, even more updated. And then you have alpha missense, and there are I would say I mostly use the first three, but I'd say the first two are probably the ones that every person tend to relate to. While for non-coding clients, as you see, we don't have so many prediction programs, but I later will introduce Splice AI, which is at the moment the gold standard for prediction in splicing for non-coding variants. So again, the take-home message is that there are many predictions. None of them is perfect. So you need to still, the confidence towards the deleteriousness of a variant is also comes from the combination of all of those. And if you see, like in this case, in our platform, it's red for any prediction program that the score goes beyond the threshold. So it's like a combination of all those gives a better answer together with fixing data. You see again the nomad V4, there are no homozygotes, so it means that this variant is not, like it's rare and found in homozygosis in patients. But at the moment, we are also moving towards a phase in which you don't have to anymore looking at single one by one at all the prediction programs. You can actually move on from an individual to a comprehensive variant predictors. And these are, let's say, the comprehensive predictors that we tend to use during my analysis. One is called Franklin from Genox. The other one is Varsom. The difference is that Franklin is fully free, while Varsom it's free if you do a small amount of search. But if you plan on using like this Varsom more consistently and more like frequently, they might ask you to subscribe. But for a few checks, it's still free. And as I said, like these two platforms are I think are very useful because for each variant, they will combine all the prediction programs, or let's say the majority of the most reliable and mostly used prediction program, so that you could have in one go a general overview of the prediction for your VUS. And finally, because we are in the world of AI, and AI is up and coming, up and running, I just want to give an example of these recent AI tools called VarChat. So VarChat is basically like an AI-related platform in which you will add your the VUS of your choice, and it will provide all the information based on literature mining. So this is like, again, an upgraded of the literature mining that I discussed before. And I just want to give you an idea about the ABCA4 variant we discussed before. So the -10 T2C, like as long as you type the variant of interest, it already gives you a description of the gene, and it already gives you like the state of the art of the research of this of each variant, and also like besides study, we'll also tell you that in what models the variant has been investigated, for instance IPS, whether there are therapeutic strategies. And this is also important because when you want to study a VUS, you also want to know whether the gene somehow has been already targeted and chosen for clinical trials or preclinical study. I think it's very important if you want to wisely direct your research as well, and funding as well. So I would like to conclude the first part of the talk talking about functional assays. So the main functional assays, it's hard to have a comprehensive slide in which you mention all the functional assays, but for this session, I think we need to at least focus on the main assays. So a VUS could be a coding VUS, could be if it's coding and it's missense, then if the gene in which the VUS locates is an enzyme, you might try to perform an enzymatic assay. Instead, like it's falls in a gene that is a transmembrane protein or like any other cytoplasmic protein, you can probably perform immunohistochemistry and try to see the localization of let's say of the protein wild type versus the protein with the missense mutation. But these, I confess, that are very, let's say, laborious type of enzymatic assays. So a very laborious type of functional assays. So it really are not so common. Okay, while for non-coding variants that are in the 5' UTR, so promoted, as I showed before, like it's possible to have a luciferase assay. And this is relatively cost-effective type of assay, and overall, it requires multiple repetition, of course, and the choosing of a proper cell line, but I think overall it will be a good way to test promoter variants at reduced cost. For 3' UTR, the story is different because you can actually measure the luciferase activity as like and measure that, but you can still measure the script level. Okay, so you will measure the endpoint result, and this can be done by quantitative PCR or RT-PCR. Really depends what type of material you have, if you have like a patient material or if you want to do this study in construct plasmids that you are introducing in cells. And finally, I will focus on non-coding variant because I say they are the majority, and non-coding variants which are not in 5' or 3' UTR. So just non-coding that are within intron or deep in the intron. And for those, we perform splicing assay. But as I will show you later on, the splicing assay is not useful only for assessing the potential of non-coding variants, but also of a specific subset of missense and synonymous variants. And this because the splicing is a very complex mechanism, like the pre-mRNA splicing affects multiple signals in the gene. So this is the cartoon. You will have the example of two exons, exon 1 and exon 2. And as you see, like the first two positions after the exon, before the following exons, these are called canonical positions, okay? And these, when mutated, these two are always giving a splicing defect. In fact, as you see, like in this is the consensus sequence. So the probability of finding a G or a T in this position is equal to 1. So there's always a G and a T. While in the position immediately before on other, you might have predominance of some nucleotides, but also presence of others. So it's like it's more flexible. While in position +1 and +2 and -1 and -2 is not flexible. It's quite rigid. So when mutated, you will have a splicing defect. Also, other positions are important, like there are either exonic splice silencer or either exonic splice silencer or intronic splice enhancer, and the balance between silencers and enhancer actually will give an estimate of like exon inclusion or exclusion. So it's like, will the splicing occur or not? And we are really aware of all these enhancer and silencers. So usually we do discover those by finding a VUS in a patient, and maybe it's it's just the only variant that remains after ring. So we functional validate and we detect whether or not there's a splicing effect. So again, like in this cartoon, I will also show that if you have 3 exons, one of the major splicing defect is the skipping of one exon, but you might also have like short, like reduction or elongation of the exon, depending on where the mutation occur. And finally, you might have a mutation that includes a segment of the intron, and therefore generating a cryptic exon. Okay, so normally these are just the regular exons, but if your mutation triggers the creation of either an acceptor or a donor of splicing, like a donor or acceptor of splicing, you might add incorporation of a segment that is recognized as it would be an exon. I just, this was for me studied extensively during my PhD for ABCA4. And what I did, I create like a library of clones in which I was mutagenizing this each and every splicing variant found in or potential splicing variant found in our patient. And we test the the splicing defect, we observed that out of all the variants that we test, a majority, those one in the box, were actually giving no splicing, no correct splicing at all. So they were all classified as very severe variant. While instead you will have a percentage, which is actually, let's say, from this variant to this, in which the skipping was occurring, but at different extent, and therefore those variants somehow classified it as moderate to severe pathogenic. But we also managed to rule out variants, although they had some prediction, in the end they resulted wild type. And it's also nice to see that, you see, like in this, for this variant, this is another -10 variant. This variant was giving no splicing defect. You might another position -20 to see giving like a moderate effect, and you might have our own -C, the one I explained before, which gives a double skipping effect, delta 39 and delta 39 plus 40, and that is a very severe effect. So that's why we need to test the variant, because depending on, it's not only the change itself, but it's also the genome test. And I also want to mention that these variants are important, especially the one deep in the introns, because like those new segments, those new cryptic exons that are formed can be reverted by using antisense oligonucleotide, which are molecules that are binding at the position, at the mutated position, and prevent the incorporation of those exogenous cryptic segments within the regular transcript. So therefore, they are a relatively safe therapeutic strategy to avoid aberrant splicing. And in fact, they are promising the targets. I don't think I have enough time, so I will just briefly mention Splice AI. So here is a cohort of cases carrying like mutation in this gene, CFAP410, and we observed the presence of this +91 variant and +3 variant. So we decided to test the effect of prediction of splicing using a new tool called Splice AI. So here you insert your variant of interest, and the nice thing about Splice AI that it gives you a score over 0.20 is considered threshold for pathogenicity. And I see like for the +3, Splice AI was giving like almost, because the range goes from 0 to 1. So 0.8 is a super high score of donor loss. Okay, so basically it was saying that a +3 with a position like here, like you have +1, +2, and there will be a +3, the donor will lost. Okay. And this was actually like a high hint for splicing effect. While for the +91, which is not close to the neither of the exons, it was nice to see a double combination of scores, either acceptor gain and donor gain. And this will tell you that basically the effect was this one. So was a cryptic inclusion. So you not only have an acceptor gain, but also donor gain, and therefore the splicing will not be anymore like as the wild type splicing, but will be aberrant splicing and introducing the exon. And of course, these were predicted to be severe, but then you need to test it. And while we will test them, we were able to confirm this. Indeed, for the plus 91 A, which is the reference, you do not see any cryptic exon, but you do see it with an A to G, okay? And for the plus 3 variant, the delta X, like the skipping of the exon was predicted due to the loss, which is this one. So this is the donor position. And once you have the T, the scores that predict the likelihood of spot splicing are suppressed, and there will be a reduction. So like it will be using another region to perform splicing. So in this case, actually, we were able to confirm both of them. I don't think there's time to continue in the second part. The second part is just the way currently I'm working on not testing this variant one by one, but testing them in a high throughput way. But to conclude the part one, I would just say that multiple predictor and database exist, none of which is perfect. And we actually aim to a comprehensive predictors over individual ones, and the more the better. The AI will help us in here because it will be able to integrate all the information coming from the web. And all these things, for how useful are, they cannot replace the actual experimental work. Okay, so the experimental work is at the moment the gold standard, and it's the only way that could, like say, fully confirm and characterize your variants. So giving the large number of functional assay available and giving the possibility of high throughput for some of them, I do predict that in the future we'll be able to reclassify and characterize most of the VUS. So I think I said that there will be no time, but I just want to show you a slide also because I was helping Tanmaji with the analysis of some of her VUSs. And what we do, basically do an high throughput splicing assay. So we collect all VUSs from our collaborators and all the people that want to send us VUS. We collect them in one library of splicing construct. So we generate the wild type construct, the variant, and the construct with the variant. So in combination of two, and we collect them all in one library, which I will just give you so this is the plasmid. We insert the segment, the tested segment, with or without the variant that you would like to test in this construct. So in between two G exons, and we will transfect it into cells, and we will see that basically like an exon is included, the GFP won't be reconstituted, so you will see red cells, red fluorescent cells. While if the mutant does not create any inclusion of any exon, P will reconstitute, so you will have green plus red, and you will have yellow fluorescent cells. Base of a combination of massive parallel cloning and FAC sorting, we will be able to test what of these variant are likely to have like a defect on splicing. And I just want to also mention that for now we were able to to test approximately 2,288 rare cases, almost 1,000 of which were in IRD genes, but we are also testing all Mendelian genes. Like if you are interested in genes that are not related to the eye, we are still testing those because the splicing effect is a general mechanism. So we are testing all VUSs that are amenable for splicing defect. And here you can see that most of the variants that we analyzed in our high throughput splicing HTSA are actually concordant with the prediction performed by Splice AI. But there are still 146 variants analyzed that gave us a splicing defect, which were not predicted by Splice AI. And therefore, I repeat myself again saying that all these prediction tools are very valid, but yet at certain point, if you do find this VUS like frequently in your patient population, you might suspect that that variant does something, and if it's not, you would like to test that. And with that, I would like to thank my group, Dr. Bujagoska, which is my at the moment, and our team members, Miyoko and Egle, and all the other members who contributed to this work. I just want to mention that if you are interested in nominate your VUS for splicing, please feel free to send an email either to Kim or to myself, and we will be happy to help and try to figure it out how to include this in our library. And thank you for your attention.
Yes, thank you, Riccardo. That was a lot of knowledge. I think very helpful, especially the AI tools and the predictors. I think it's something that all of us can do on our own if we get a VUS. So Yes, so I have a basic question to Riccardo also, and anyone else can answer it. Now, if we get a VUS and based on these predictors of Splice AI or Revel or CAD, we just find out that it is likely to be pathogenic. How does it translate into counseling of our patient, or how do we get it to be formally or officially reclassified? Like, what is the procedure? If anyone knows, like, how do we go about that?
Yeah, so this is something that I would like to, I don't feel like giving like a conclusive answer because, I'm not trained as genetic counselor, but I would say that I would probably highlight this variant as a potential like a culprit variant, but I would not feel able to or confident of giving any answer to my patient unless this variant is not studied. And for instance, if I do ended up looking in to an article in which the same patient has been with a different mutation has been associated with disease, in that case, I would say to my patient that, hey, it's likely that this is your second hit and your second variant based on this functional lesson. But I think at this, today, I don't think people will, it will be easy, it will be fair to the patient, this is your causal mutation if you don't have any proven functional study. So I will keep it at the will be strict and just keep it at the US level until it's not solved. But it's a dynamic process. So especially with these AI tools, might, maybe in 3 months, we have been reclassified many variants just based on the fact that we're working on that, And I think in our field of genetics, like the collaboration really helps because you might actually discover that groups are working on the same genes and making the same efforts. And, hopefully your variant will be reclassified at a certain point.
So, thank you, Riccardo, for answering that. I had a similar question. In fact, many a times when we test our patients and we get these VUS and we do have, a patient who wants a, a prenatal counseling or something like that, where, the decision is based on the genetic variant that we find. And unfortunately, if it comes as a VOS, then we are stuck, like we can't go ahead any further and counsel them regarding that. So as you're mentioning that only applications of these, the first four or five of the options that you showed, including the AI, the various databases, this is not going to confirm it or convert the VOS to a likely pathogenic or benign, right?
No, I use them, let's say, I use them to prioritize my studies.
Mm-hmm.
Okay, so for instance, in the case of TMEM, sorry, we and other colleagues of part of that article, we observing that TMEM variant so frequently that initially we were like, okay, this is benign.
Mm-hmm.
Then by analyzing like more in depth the ethnicities and by excluding the presence of variant in Europeans, which is like, more than 1 million alleles,
Mm-hmm.
we were like, okay, now we need to do something. And since it's like a 5-star, we had to do like a promoter analysis.
Okay.
But in other case, for instance, with the intronic and deep intronic, we do observe like the splice, the ABCA4 minus 10 variant. If we do see them in patient so often, then over controls over general population, then we prioritize that variant for investigation. So the same in your population, if you have like like groups, like genetic isolates in which you see like that all like large pedigrees or large families, they keep having like recurrently this variant, I will definitely hold on this variant and putting in in the bucket of the VUS that are likely to be pathogenic.
Okay.
But you do need to test them,
Okay.
unfortunately.
Okay. Mm-hmm. So after the functional
after the
assays, then how do you change it to likely pathogenic or pathogenic? Like you
Yeah, so
report
in
these, or you go to the various databases and change it? Like how do you change it?
the way I usually change it is, and I'm happy to give these more details another time. Unfortunately, you see, there's no time for the detail on everything. But the
I
way I
know.
normally do is, I use the American College of Medical Genetics
Mm-hmm.
guidelines, which is 2015 published article that now has been also updated. Okay, so it's like these ACMG guidelines, let's say, were released in 2015, but now there are, more sophisticated type of classification. Okay, so one of them says that. So it basically like it's a sum of different scores. For
Yep.
instance, is your variant rare in patient population? Yes, and you add some score, some points. Is your variant present in all affected and follow segregation? Yes, and you add some scores. But like is predicted to affect a known enzymatic domain, a known domain is predicted to affect the same position and as other variants in previous study, and all these type of things. At certain point among the request, there's like, was your variant validated through a functional assay? So, and if you say yes, it kind of rise the score so much that it passed the threshold for significance, and then it will pass from a VUS to a pathogen, likely pathogenic. Usually, it's a likely pathogenic.
Okay.
Okay, of course, if your VUS is a stop codon, so I just want to clarify. When talking about VUS, I mostly refer to either missense variants or non-code variants.
Putting
Because
variants. Okay.
when you have like a +1, +2, -1, -2, when you have frame shifts, when you have codons, usually they already hit the threshold of severity, because among the criteria, one of the first questions for severity is your variant a stop, nonsense, loss of function? If you say yes, you're already getting a quite amount of points to hit that significant level. Okay, so if you have a stop and you just say yes for being a stop variant, plus it segregates in your families, plus you have multiple instances of that variant in other patients, that is already enough to classify as a severe variant.
Mm-hmm.
Okay, but if instead of having a +1 and +2, you have a +3, or you have a
Mm-hmm.
-10, or you have something +91, or you have something in motor, for how much you want to populate this scoring with all these insights, you still need the functional assay because you cannot otherwise prove the severity, right?
Okay, got it.
Yeah. So for instance, for the TMEM promoter variant, right? No coding. So you cannot say anything about predictions in coding. You cannot say anything about you know, enzyme activity, anything. So the only thing you need to do is just check in expression, and you have to do it through a functional assay. But if the
That's right.
expression is decreased, and you have done this experiment with so many replicates in human patients and everything, is so important that
Mm-hmm.
would give you like the significance for moving to pathogenic. Yeah, unfortunately, it's frustrating because for many variants in our studies, for how many, let's say, insights we collect, once they are missed, and we cannot do those laborious immunohistochemistry or those laborious like cellular assays that are
Mm-hmm.
different from a splicing assay, we still need to say, okay, we were not able to do a functional assay, and therefore our variant will stay a POS.
Mm.
Although as a, although by being a missense has been predicted by multiple prediction tools as deleterious, you know?
Mm.
That's what's happening with most of my variants, I think. I mean, yeah, that's a topic for another time.
Yeah. So like, I was helping Tamaji with so many, with some of her VUS, but they are missense variants. Okay. So when you test them through Splice AI, their Splice AI score doesn't hit the significant threshold. Yet I decided to test them, and for few of them I have the results. I didn't show it this time, but for few of them I have the results, and there's no difference between wild type splicing and mutant splicing. For I say to the machine, this might be likely pathogenic based on family history and population and everything, and rare like an allele frequency, but we cannot prove them through spring at the moment. So you should aim for a more sophisticated way of testing them.
Suppose one more question, I think probably. So suppose we show by segregation analysis that the particular variant is present in all the members who are affected in the family and absent in all the members who are not. Is that enough to then reclassify the variant?
No, it's a very, it adds a lot of points to the classification, but unfortunately it doesn't pass the VUS. So for instance, I think if you all agree, like maybe another time, I can just discuss, I can send around the article to Tamaji. Tamaji will distribute it to everyone, but to everyone who's interested. But next time, for this next use of time, it will be nice to just show in an interactive way. I do classify this, like I do have it like in this article that I will send around. There are already all the criteria, but then we can have a couple of examples on real families and real assays. But no, unfortunately, that's the frustrating part. Like that is like a big thing, right? It's present in sometimes we have variants that are the same exact variant in 3 families, all of which have your situation, right? Well segregated within affected that are excluded in unaffected, and yet you cannot say. Unless you do not have like say 100 family, like you don't have such a high statistical power, which which you have like 7 families, 8 families, like large pedigrees that follow your, the above mentioned criteria, then in that case, with the new guidelines, it will be, it will add more value. But honestly, I need to talk to my genetic counselor. I don't know if that is enough, just that to add significance.
Mm-hmm.
It's a good question. I will take note.
Can I ask a question?
Like how many families, you know?
I'm sorry, we are shooting the time, but I mean, if do you have time for another question, Ricardo?
Yeah.
Yeah, Ria, go ahead, please.
Okay, so like you were showing us the OMIM database, so some of the genes, they will have AR/AD written in front of them rather than one inheritance pattern. So I understand that maybe that gene works in a haploinsufficiency kind of manner and dosage effect and all of that. So that's why it follows both. But we often get reports from the lab with AD or AR, one of them written. So how is the lab choosing for my patient, how will it sort of segregate? Especially, I mean, it's especially for times when it's matching with the phenotype. So I want to consider it, but I don't know which, whether it should be homozygous or heterozygous.
So I would say OMIM gives you just the all what has been found, right? And then we have, like, for instance, we study retinal degeneration. So if we have a syndromic, if we have mutations in a syndromic gene, uh, the first thing that we try to do is actually go by the patient and just make sure that this patient doesn't have any syndromic feature, right? So if they have no syndromic feature, and let's say keep doing the job of classifying, and only if we accumulate enough patients that have the eye retinal only phenotype, we are able to somehow pull the data together and try to aim for a publication in which we say, look, this gene not only gives syndromic, but also give non-syndromic. So the way you choose is actually, let's say, you can pick from the report your most interesting VUS, but if they are on a syndromic gene, to claim that they are responsible for your eye phenotype, you should actually reach out to your colleagues and say, hey, we have this family within. Have you have instances of similar phenotype genotype correlation? Because sometimes what happens, and again, there are many cases, I'm happy to share those another time. There are cases in which you have a specific portion of the gene that are more, let's say, when mutated, they give a syndromic phenotype, and are more tolerant. So when mutated, they give to eye only phenotype. But it's hard. Like, the only way to do it is just to share knowledge with your colleagues, and you cannot claim this based on just one family.
Okay, thank you.
I think it's important to stay unbiased, though, and not include things just because, they were found in syndromic genes, because the more we sequence, the more we are discovering novel genotype phenotype correlations.
Also, is there a criteria to submit the BUS to you, in the sense that maybe it's just the lab that we are getting it done from and the databases it has access to, that's classifying it as a BUS, but actually it might be something else? Do we need to go through a process before we send it to you, or?
Well, I would say, let's say, tools that I just, shared today, there will be a huge pre-filter for, for sending a variant. Because if you start to say, hey, you know what, these variants are all in the South Asian population, so say India, Pakistan, the subcontinent, not present in Europeans, segregating in the families, apparently rare, and this and that, and you have few of them with the Splice AI score, like, again, as I said, like, giving the high number of variants that we receive we ask all the collaborators to have a sort of sanity check and pre-filter, because ideally you like to study them all, but it's very hard to, for it's labor intensive when you are dealing with thousands of variants at the same time. Or high throughput, you still have some validation steps, that might require like more time. Okay, so I think it's important to do all this pre-check, and then we are very open to discussion. If you're fully convinced that this is the end and will solve the family, uh, if possible, will be included. Okay, it doesn't have to check all the boxes. It has to be just a fair number of boxes. But yes, the SPI is probably one of the scores that we would like to see, that it just is above the 0.2, that gives more confidence in testing the value.
Mhm.
So anyone who's still at the chat, any questions?
I don't think so, Rhea. Maybe we can conclude now.
Okay, thank you so much for today, Dr. Riccardo. That was extremely useful. I would like your permission maybe later on if we can share this in our ocular genetics interest group, the recording, so that in case we miss certain points, and only if it's okay with you. And yeah, and I would also want to be in touch for probably a follow-up, because it seems like there's a lot more to discuss still, and we can plan it
Okay.
for another time when you're free.
Unfortunately, I didn't have the chance to go through the high throughput part, but I thought it was necessary to just to give like a first layer, because there are so many things, and these are all needed to go through in the second phase, which is the high throughput. So, but I'm very happy for the opportunity. Thanks again, Tanuji, for for the opportunity. I really enjoyed to be here and to discuss this with all of you, and looking forward for new opportunities to collaborate. Yeah,
Thank
Yes,
you.
thank you. Thank you for agreeing to speak to us.
of course, my pleasure.
Yeah,
Thanks.
thank
I
you.
know
See you
that,
again.
bye. Bye. Bye.
Bye. Bye.
Bye.
Bye.